All right. Just to recap, what we did yesterday is we tried to build a normative theory of neural computation, starting from an objective function and then optimizing that objective function to obtain an alternating optimization algorithm, which both computes the output of a neuron as a weighted sum of the inputs and updates the synaptic weights so that the total reconstruction error is minimized. And that, of course, corresponds to performing principal component analysis on the input in the online streaming setting. And then we compared this result with experiments and found some encouragement. But when we tried to extend it to multiple neurons, to multiple principal components, we ran into a problem because the learning rules were non-local and therefore not biologically plausible. So we looked for another objective function that could give the same learning rules for single neuron, but local learning rules for multiple neural, multiple component PCA. And that was the similarity matching objective function, where we minimize the mismatch between the similarity of the inputs and the similarity of the outputs. And when we did that, we obtained what we wanted, which is multi-component PCA with local learning rules. So that was very satisfying for us because, you know, after so many years, there was a solution that is both normative, that is related to a principal objective function, but also biologically plausible. But perhaps what was less than satisfying is that a principal component analysis is just a linear dimensionality reduction. And so the problems that it can solve are really very basic. There aren't that many interesting things that you can do with it. And as we know, the brain excels at solving problems that require nonlinear operations. Even neurons themselves are nonlinear devices because they produce a non-negative output. They have a rectifier in them. So confronted by this disparity, what we decided to do, and that's what I'm going to tell you about today, is to get biologically, even more biologically plausible algorithms. And the first one I'll talk about is the one which requires that neuronal output is non-negative. And the second one takes into account another biological piece of knowledge, that neurons actually have multiple electrotonic compartments, and I'll explain what it is. All right, so let's move on. As before, I would really appreciate if you ask me questions in the middle of the talk. If you don't understand anything, feel free to interrupt me. And before we move on, I wanted to ask if there are any questions about yesterday.
No questions yet.
Okay. All right. So let's move on to, sorry, did I go back? Oops, I'm sorry. Was it the wrong place? Here. Okay. So, so what we did was really very, very basic, but it led to some really unexpected and very interesting results. We looked at the objective function. And as I said before, it's trying to match the similarity of inputs and to the similarity of the outputs, okay? And then we said, okay, well, but we know that the neuronal output has to be non-negative, right? Because if the neuronal output is described as a firing rate, number of spikes per second, that cannot be a negative number. If you have graded potential neurons which don't spike, then still the rate of vesicle release from the synapse is also a non-negative quantity. So neurons seem to be really acting as rectifiers. So what we did, we just added a non-negativity constraint to our objective function. So that's all we did. And then all hell broke loose, unexpectedly to me, because I actually didn't know that such an innocuous change in the objective function would have such major ramifications, because the solution of this objective function is no longer PCA, no longer linear dimensionality reduction. And what it is actually depends on the data set. So let's first look at the simple case where the data set is of the kind that I showed you in this experimental data, remember, from the human fMRI recordings and the macaque monkey electrophysiology, right? They measured activity vector elicited by different visual stimuli, both animate and inanimate. I'm simplifying that big, colorful matrix that we had before. And they computed the correlation between them, or in the product. And there were two blocks, if you remember, because the activity patterns in the brain in response to animate stimuli were more similar to each other than between animate and inanimate. Okay, so what if we use these neuronal activities, right, taken from the brain as axes, and we will give that as an input to our algorithm and figure out what y is going to be. And just as we did before, remember, we restricted the dimensionality of y, we made it smaller than x. So let's do the same here. Let's take that y is just two-dimensional, okay? Then what essentially we are asking the algorithm to do is to approximate this similarity matrix that is shown in colors by a low rank 2 matrix in the best possible way. Now, what's going to happen if you were to do that? Well, red squares are negative. We cannot have a negative correlation, negative inner product, if y's don't have negative components. So the best we can do is to have them set at 0, okay? And then the blue blocks, because we only have rank 2 matrix, can only be a constant matrix. We will set it to 1, okay, because the diagonals are 1, so it kind of makes sense. So this is the best possible approximation of the matrix on the left with a rank 2 matrix factorizable in non-negative components. As you can see immediately, what we achieved is a clustering solution. We basically grouped all the animate objects into one cluster and inanimate objects in the other cluster. And to make this really intuitive, I also show in here the activity of the neurons, the Y neurons. There are 2 of them. One will be so-called animate neuron, and the other would be inanimate neuron, okay? And so animate neuron has this pattern of activity. Inanimate neuron has that pattern of activity. And then when we compute the covariances of the 2-dimensional activity vectors between each pair of stimuli, then we get this block diagonal matrix of 1s and 0s. Okay, so what I'm saying here basically is that if you gave the input on the left that we got from fMRI and electrophysiology in the brain, then the algorithm would classify that input into 2 categories, animate and inanimate. So instead of doing PCA, by introducing the non-negativity constraint, we achieved clustering, classification. And that's a big deal because now we can use this classification to make decisions, right? Should you, should you fight or flight or whatever, you can make behavioral choices. Now, this is, of course, what we call a solution in the offline setting, when we have the whole data at once. But if we were to have this algorithm implemented in the neural network, we would have to do it in the online setting. But we're already professionals in doing that, right? We can follow the same prescription as we did before. We start with the objective function. We massage it by transforming this cross term into a form where there is a covariance, there is a sum of outer products, which is a covariance, and we use that covariance from the previous step, just like before. Then we can iterate to find the optimal y. The only difference from the similarity matching PCA network is that now we have to project the result at every step to the non-negative sector, right? So we just cut off the negative solutions and set them to 0, okay? And that actually works. And this can be accomplished naturally by the same network we had before with 2 output neurons. This is our animate and inanimate detector neurons. And the only difference, again, the blue links are this, the weights of the synapses in the blue links are given by the components of the matrix WYX, and the red links are given by the components of the matrix of WYY, just like before, but neurons are rectifiers. So they compute a weighted sum of inputs. If it's positive, they transmit it. If it's negative, they set it to 0, just like real biological neurons. And then we have the learning rules, which are exactly the same as we had before. They're local. That's why there is this happy face icon. And using those learning rules, we can learn the classifiers that would decide whether the stimuli we are seeing are positive or are animate or inanimate. Okay? Any questions?
No questions yet.
Okay. All right. So, okay, so I gave you an intuitive idea that this should be a clustering solution. We derived a neural network, both including activity dynamics and learning. Now, can we actually test that this is a viable clustering algorithm? Like, can we compete with clustering algorithms that people have been using in machine learning? In fact, we can. Here is an illustration. So in this case, the data set is just 2-dimensional, x1 and x2. And the points come from 3 classes, red, green, and blue. And they're presented to the algorithm sequentially. That's the online setting, in the order shown by the number of the points. But of course, the algorithm doesn't see the color of the points. We just kept the color to remember which point came from which cluster. But as we throw these data points at the network actually classifies them almost perfectly. So this is the output of the network. And as you can see, it shows the activity of the 3 output neurons. We will call one of them blue, one of them green, and one of them red. Doesn't matter which one is which. There's spontaneous symmetry breaking. So the first 5 points elicit the activity of only the first neuron. The second 5, the second neuron, output neuron, and the last 5 points elicit the activity of the last third neuron. In fact, this is what clustering or classification is supposed to do. There is an interesting situation with the point number 13. which elicits the activity in both red and blue neurons. So they can decide which cluster it belongs to, right? The red and blue are sharing that point. But I don't see it as a bug, I see it as a feature, because if you look at where point 13 is, it's actually halfway between the red and the blue cluster. And because the algorithm doesn't see the color, it has no way of knowing whether this is the point that was generated by the centroid of the red cluster with the noise pushing it towards blue, or the other way around. And so it decided to indicate this uncertainty by attributing that point to both clusters. And this is so-called soft clustering. Okay, so the network works, and also you can see how it works, right? That you get an input X and a vector X, and it has two components, and those components are projected to each of the three neurons that are classifiers. And then what happens is each of those neurons compute the weighted sum of the inputs. But the weighted sum is just a projection of that vector X onto the centroid represented by a given clustering neuron. And so the neuron whose centroid is closest to the data point has the highest output. And then it starts competing with other neurons in this recurrent computation here, okay? This circuit is slightly different from the one we had before, because before we had direct inhibitory connections between neurons. And here we decided to get a little bit more biologically realistic and had intermediate inhibitory neuron. But it doesn't matter. It's a slightly more complicated math, but still derived from the objective function. Anyway, these neurons are competing with each other, and the one that had highest correlation with the input data point wins, and it suppresses all the other ones. And that's why the result of this algorithm is a clustering output where only one neuron is active and the other are silent. So that's basically how it works. Any questions?
Questions? No questions yet.
Okay, so as I said before, we don't really, what this algorithm, the non-negative similarity matching algorithm outputs, actually depends on the data. So in this case, we used a data set which is easily clusterable, right? 3 clouds. So the algorithm clustered it. But what if we throw it at a data set which is not easily clusterable, right? Something like natural images with which you started yesterday. So you throw a data set which consists of little, remember, little squares which are cut out of natural images, where we took each square and we vectorize the pixel intensity in that square, and we throw it at the algorithm. Let's do that. And as a result, it turns out the algorithm learns the feature vectors for each neuron, which are edge detectors of various orientation and location in the visual field. So each of those little squares here are is the reformatted synaptic weight vector for WYX for the inputs to each of the 25 neurons here, okay? So it's interesting that the same network that we use to cluster for clusterable data can also compute the feature features that are observed in the brain, in the visual cortex. Okay, sorry. Okay. Just read in the chat. So now that we were able to actually learn edge detectors, which are the features of the neurons in the primary visual cortex, we thought that maybe we should try to tackle the big problem of how visual processing happens in a human or other mammalian brains. And there, as you know, the input comes in a pixel form. This is light intensity captured by the retina. Then it's transformed through a couple of steps into representation in V1. That's where edge detectors are. And if we compute the similarity matrix for here, we should be computing it for those features, right? So if you present edges of different orientation and compute corresponding activity vectors, then this is the similarity matrix you get. It goes through 360 degrees rather than 180 because actually the experimental data we used from the Chiani lab was for moving edges, okay? And so the vertical edge here is say moving on the left to the left, and here it's moving to the right. But still, you can see the pattern of similarity that arises in this cortical area. Then what the brain does, it passes signals through consecutive areas, V2, V4, and so on, come into the area called IT for inferotemporal cortex, where this fMRI data and electrophysiological data that I showed you before was obtained with this big similarity matrix. And you can see that in the course of those transformation, the similarity has transformed quite a bit. And so the question arises, if our basic sort of primitive computation was similarity of the input is similar to the similarity of the output, then if every layer preserves similarity, how can we change the similarity so drastically? And this is, of course, because the only part of the objective function is trying to preserve similarity, right? We also have a non-negativity constraint, which prevents us from matching the similarity exactly. Sorry, I just saw someone's question in the chat. What was Z on the central graph?
Just this picture.
This picture?
Yeah. Is it meaning of Z?
Let's see. Oh, okay. Sorry. This is the thing that I glossed over. So, so when we replaced, this is a very good question. When we replaced the direct inhibitory connections implementing competition between different channels with the indirect connections going through the interneuron, Z is the activity of this interneuron. Mathematically, it comes out of implementing the constraint that the total activity of all neurons is no greater than 1. And we put this constraint into the objective function using a Lagrange multiplier. That Lagrange multiplier is Z. And so what this inhibitory interneuron represents is the Lagrange multiplier for the constraint that you need to do clustering. Does that help? Okay. Sorry, I'll keep an eye on the chat better. Okay. So basically, the issue here is that, well, what's going on? Is our objective function sufficient to change the similarity so drastically that you go from the similarity in the pixel space to the similarity in the edge detector space to the similarity in the object space? And we tried to push this as far as possible, and I think we can partially reproduce what is going on. So to explain how that works, I want to give you a way of thinking about visual processing using high dimensional spaces. So why high dimensional spaces? Well, because if you show an image to a person or an animal, image say has 1,000 by 1,000 pixels, and then the pixel intensity of the image can be thought of as a vector that lives in a million dimensional space. These high dimensional spaces are very difficult, at least for me, to imagine and intuit about them, but I'll try, okay? So suppose you have two individuals, right? Sam and Joe, but you have many images of each person taken at different angles and different illuminations, maybe different facial expressions. So because for the same individual, you can continuously transform one image into another by slowly turning the view, for example, or changing the expression or illumination, all those images live on a high dimensional manifold that is embedded in even higher dimensional, this million dimensional pixel intensity space. So what I tried to show here, well, it's of course a projection on 2D, but what I was trying to show here is in blue, the manifold of faces corresponding to Sam. And in red, the manifold of images corresponding to Joe, okay? What I was trying to demonstrate is those two manifolds are highly entangled, right? Because, you know, a view of Joe from one direction, say face on, could be rather similar to the view of Sam from the same direction. But two views of Joe, say face on and profile, could be very different in the pixel space. And so that's why there is this entanglement, right? So the manifolds corresponding to two faces can come closer to each other than the points on the same manifold. Of course, those two manifolds never intersect in the high dimensional space because these are two different faces. So there should be some difference on the pixel level. But because I'm only drawing a projection in 2D, it's hard to demonstrate that there is no intersection. But of course there is. So some people suggest that to solve, for example, an identification problem where you classify the images, whether it's Joe or Sam, reduces to pulling those manifolds apart without breaking them. That allows one to classify them simply with a linear classifier. And so the idea is that that was this deep network is doing. It's taking these manifolds and it starts to pulling them in separate directions until they don't entangle at all. And then once they're pulled apart, you do a linear classification and you classify them perfectly. And you can always say which image is Joe and which image is Sam. Okay, so this is sort of manifold embedding view of image classification.
Can I ask you a question?
Yes, please.
Could you repeat, please, what are the points on these manifolds of representation of human faces? Are there a combination of impulses from different neurons?
Yes, so on the very first, so it depends which layer you are on. And the very first layer, this is just pixel intensities on the monitor that shows you the image, okay? So as before, you take pixel intensities, there are a million pixels, so you reformat them into a vector, and it's a million dimensional vector, and I'm trying to plot that vector. Now, as you go deeper in the brain, these are now activities of the neurons in that layer, corresponding to that particular image. So given image elicits the activity in each layer, and then in each layer, you can form a vector of activities, and that vector is represented here as a manifold for many different images. Does that help?
Yeah, thank you. And how do you entangle these two manifolds? Just linear transformation?
How do you disentangle them?
Yes, yes.
Oh, so that's the big problem. So that's like what all machine learning is about. So most of the data that we get in the world comes from the high-dimensional entangled manifolds. But to solve the classification problem, to say this is Joe or Sam, you cannot really classify those entangled manifolds, sorry. And so you have to pull them apart until you can classify them linearly.
Okay, thank
And
you.
you learn, yeah, sorry, what?
I just say
Sorry.
thank you.
Oh, okay, yeah, okay, great. Any other questions?
Yeah, one more question. What kind of geometry do you apply, have in mind when you consider embedding in high dimension? For instance, usually when you try to embed network in high dimension, it's useful to consider hyperbolic geometry. And here you have just a standard geometry in high dimension or not?
You're talking about the metric?
Yes, to some extent, yes. So is it just a standard Euclidean matrix in high dimension, or you use a kind of hyperbolic geometry?
So that's a really good question. And I actually don't know which one should be using. We're just using the regular Euclidean metric, but it doesn't mean that this is the, I mean, certainly not the only one, and certainly it may not be the right one. So yeah, so we only tried Euclidean.
Okay, thanks.
Okay, so this is kind of an intuitive picture from a review of DiCarlo and Cox. Also, Lee and Sung had a similar intuition. And the question is, can we achieve that with our non-negative similarity matching? We tried to do that on a simple, on a simpler, oh, sorry, what's happening? Okay, sorry about that. Okay, so we tried to do it on a little bit simpler problem where the data is only two-dimensional, okay? And the data points live on two manifolds, right? The two little spirals. And you cannot classify this data into two spirals by a linear classifier because they're entangled, right? So the question is, well, can we do this with our network using this intuition of manifold disentangling? And we tried to use a network which is a variation of this non-negative similarity matching network. It's a slightly different objective function that I write down here. But the reason why it's different is rather technical. For example, one of the things we need to do is, before we talked about describing distances by inner products, but you really want to have the Euclidean distance because, you know, the inner products don't really describe distances well if the points are on the same side from the origin. So you really need to write down the real Euclidean distance, and you also need to have a threshold to decide where you're going to draw the line. So basically, this is the objective function that we use, but it's a rather technical point. You can think of this as still being a clustering algorithm. And so when we ran this algorithm with two output neurons, because we have two clusters, this is what we got. Okay, so it's a failure, right? Because what happened is that one neuron was active only in response to this stimuli, and the other neuron was active only in response to this stimuli. And if we changed alpha, that would change the threshold, but it still didn't work well because basically there was never a situation where one neuron represented all the data points from one spiral and the other neuron all the data points from another spiral. So basically we failed, but we didn't give up, okay? And we said, okay, well, maybe the problem is that we only have two neurons, right? Because they only capture a very small part of the data set. So let's rerun our algorithm on the same data set, but with 30 neurons. And then something very interesting happened. It turned out that when we did that, the first 15 neurons shown here learned to respond to the stimuli that belonged to one spiral. And the other 15 neurons learned to respond to the stimuli that belonged to the other spiral. Each neuron still captured a small subset of the stimuli, but together they covered the whole spiral. Moreover, the manifold representation is still intact here because the receptive fields of these neurons are not completely independent. They have an overlap. For example, look at this neuron and this one, okay? They have different receptive fields, but they do respond sometimes together to the same stimulus. And that means that they are preserving the neighborhood relationship that existed in this manifold. So the manifold didn't get torn or broken. The manifold is preserved. And this is very important because, if we start breaking the manifold, then we can make mistakes in classification, because then some faces on different parts of the manifold will be clustered separate from the others. So, so this is great news, but we are not done yet, because we still have to classify this, this data into two clusters. Not 30, but two, one spiral and the other spiral. So what do we do? Well, if you allow to do some supervised learning, all you need to do is to have a second layer, which will just learn from instruction which neurons correspond to the first spiral and which neurons correspond to the second spiral. But we were determined to do this in completely unsupervised manner, without any supervision whatsoever. And so it turns out that we could do it by slowly reducing the number of neurons in this pyramid scheme, which has 6 layers. So in the first layer, we have this 30 neurons, and this is even more complicated situation where spirals are even more entangled. But the first layer already kind of separates the data from the two spirals. So if you can see here, this is a similarity matrix. And as you can see, the off-diagonal blocks are completely zeros. So the separation already happened. But what we need to do is to learn that there are just two manifolds. And this happens if we reduce the number of neurons in each layer by a factor of 2 until we get just to 2 neurons, and then they're separated. So just like in that intuition from DiCarlo and Cox that I showed you, this is what happens in our network. The two manifolds get slowly disentangled and pulled apart until they're so far that they can be classified by simple clustering algorithm. I want to emphasize that this network uses exactly the same learning rules, and it has exactly the same architecture in every layer, except that the number of neurons shrinks by a factor of 2. And yet it can perform this non-trivial classification. Any questions?
Is this phenomena related, when you consider general network, sometimes there are transition, clusterization phase transition. So instead of randomly distributed matrices, you have just clusters get generated. Is it similar phenomena? So a kind of clusterization of the general network, or it has nothing to do with that?
Maybe. So let me say something that may help to make this connection. So what is shown, let me explain what is shown in the second row. So the second row shows two-dimensional embedding of this data set from each layer, okay? But the two-dimensional embedding is done with a spectral method, okay? So if you computed the Laplacian, this would be like the two highest eigenvectors corresponding to the two highest eigenvalues, okay? So when we're doing this embedding, even though in similarity matching, you can see that the data from the two spirals separated, in the top two eigenvectors, there is no separation. So where is the separation? Well, the separation lives in some much higher order eigenvectors corresponding to very small eigenvalues. And this is why we needed supervision to classify it in the second layer. So if you could use supervision, supervision will just point the weights in the direction of the separation between two spirals and classify them. And that could be done in two layers. But because we don't want to use a supervised signal, supervision signal, we had to do it step by step in this multiple layers. And this is because we basically pushed down all the dimensions that corresponded to this useless directions corresponding to high eigenvalues until we got to the directions corresponding to the low eigenvalues where the useful information is. So if I understand correctly, at least a little bit from your talk, Alexander, so that if you use spectral methods, then you can detect this difference that you're talking about between the clustered and chaotic networks. And you can also use spectral methods to understand what we are doing here. So if it is exactly the same or not, I don't know. But the same tool would allow one to understand this.
I see. So it looks that the spectrum of similarity matrices can be analyzed in a similar manner. Yeah.
So, but I don't know if it's exactly the same phenomenon that you're talking about. But you can use the same tools.
Okay. Thanks.
Any other questions?
No more questions.
Okay. Well, I mean, as always, in the interest of full disclosure, I don't want to oversell this work. And this is probably just a nice illustration of what non-negative similarity matching can do. It doesn't necessarily tell you what the visual system really does, because this is a little bit, things are a little bit too simplistic here in terms of finding invariances. But anyway, at least we were able to show that the algorithm we developed, driven by biological inspiration, can produce rather interesting machine learning results. So are there questions? No? Okay. So if there are no questions, the last thing I wanted to talk about is again driven by the biological observation, where we try to put in even more biological realism, yet stick with this normative algorithmic approach where everything is derived from the objective function, and therefore we know what the network produces. So we don't have to guess. In most cases, what the output of the network is, we know it. And this has to do with something that I've been exposed to for a long time. You know how it happens with experimentalists. They discover something great and they keep telling you, oh, you know, I discovered this amazing thing. And as a theorist, you kind of think about it and you're like, well, I don't know how this fits into my theoretical framework. So I don't know what to do with it. And you kind of go around, and in my case, I've been ignoring this for many years. And it was partially justified, I think, because many neurons don't have this. But a lot of neurons also are like that. And eventually, we basically realized that we can have a theory of this phenomena, and that's what I'm going to tell you about. So this is the observation that the majority of neurons in the cerebral cortex, which is kind of supposed to be like the thinking part of your brain, right? The one on the surface with the folds, the subject of all these jokes, most neurons there are so-called pyramidal neurons. They're called pyramidal is because their cell body has like pyramid shape like here. But they have another interesting properties. They have dendrites, the inputs, where the inputs come into the neurons, of two kinds. The so-called basal dendrites that are here below, and the apical dendrites that are here. And those two kinds of dendrites receive two kinds of inputs that come from different places. The apical dendrites get input from downstream parts of the cortex. Remember, there was this layering diagram which we tried to understand in terms of disentangling. So the inputs to the apical dendrites come from the right of that diagram, and the inputs to the basal dendrites come from the left of that diagram. And so people kind of think about those inputs as being observations, sort of feed-forward signals, and the apical ones as feedback, maybe prediction. Perhaps Carl Friston talked about this, right? How the brain is trying to predict the observations. And so that's where those prediction inputs come in. But it's not just the two kinds of inputs. Also, what happens is that those two inputs get summed separately. So the basal dendrites sum up its inputs in a weighted manner, of course, where the weights are given by synapses. And the apical dendrites sum up their weighted inputs. And then the soma gets both of the inputs. The communication between the two so-called compartments, the basal and the apical compartment, is implemented largely by the so-called calcium plateaus. This is kind of a signal that propagates within a neuron mediated by calcium that is different from a sodium action potential that travels along the axon. But this is a real thing, and it does communicate between the two compartments, the integrated, the summed values from those two compartments. Okay? So it turns out that this pyramidal neurons is much more complicated than the little neuron we considered initially, for which we did the PCA model. So if this little neuron with just a single summation compartments, which computed one weighted sum of the inputs, where the weights are given by the eigenvector of the covariance, and then outputs the principal component. So if we can describe that simple neuron, which also exists, this is also valid, which we can describe this neuron as PCA, what is the right algorithm that corresponds to this pyramidal neuron? So that's the question. And the hypothesis that I'm going to pursue is that this algorithm is so-called canonical correlation analysis. So I know that canonical correlation analysis is not as widely known as PCA. So what I'm going to do in the next slide is to basically explain to you what CCA is in a very simplistic, intuitive manner. So if you already know, I apologize, but just to make sure we're on the same page, suppose again you have a two-dimensional data set, X1 and X2 are components, and these data points from that data set, they're going to appear online sequentially, and they will follow the hand of an old analog clock face. Okay, so I realize that the younger generation only have seen digital clocks, so that may not give you the analogy. So I'm going to show what happens. Okay, so data appears like this. Okay. And then that corresponds to the, hours on analog clock face. And then there is another data set, also two-dimensional, but it doesn't have to be. Could have different number of dimensions, but in this case, it's the same. And the data points also appear in the same order, 1, 2, 3, 4, and so on. The data points from the two data sets come in pairs. So first you get 2 1s, then you get 2 2s, then you get 2 3s, and so on, okay? And so the task of the canonical correlation analysis is to find two projection vectors such that the correlation between the projections is as high as possible, okay? So remember, for PCA, we had to find one projection direction, so the variance is the highest, okay? Here we have to find a projection direction for the first data set, projection direction for the second data set, so that we can compute the projections and then compute the correlation between them. It is as high as possible. Is this clear?
Yes.
Okay, great. So mathematically, the canonical correlation analysis is written like this, okay? So this is the objective function. You multiply the covariance of the projection of the X data on WX and the projection of the Y data on the WI. And here I'm assuming that vectors X are column vectors, vectors W are row vectors, and I'm maximizing with respect to WX and WY, subject to the normalization condition. This is simply the fact that the total energy in the X projection is fixed, the total energy in the Y projection is fixed, okay? But you have to find WX and WY so that those projections give the data which is as correlated as possible, okay? So, for example, this was a simple case where the two projections can be chosen to be the same, say in the vertical direction. And then if you project the X data set on WX, Y data set on WY, you get this cosine looking curves, and then they give you the highest covariance because this is just like cosine squared. Okay, can't have anything better than that. But if the first data set was the same as before, but the second data set now is shifted, okay? So that someone turned that clock face, okay, to the right, but the data points still appear in the same order. Ones appear with ones, twos appear with twos, and so on. So that means that if you project the first one on WX, you would have to project the second one on a different WY, which is now pointing not vertically, but towards the 12 o'clock. And you would get the same projections, and then you again get correlation, which is cosine squared, and you maximize the correlation, okay? But if you didn't realize that and you kept the WY pointing vertically, then of course, you wouldn't get the maximum correlation because the two curves would be shifted. Like here, for example, again, the data appears at the same location, but WY is chosen not wisely, okay? And so in that case, the two cosine curves are shifted and you don't get the highest correlation. So this WY does not maximize the covariance, okay? So this is not the optimal solution of CCA. But that gives us an idea about how to learn optimal WX and WY, because you might think that, hey, you know what went wrong here is that there is an error between the two projections. There is a variance, okay? There is a disparity. It goes in different directions here, but maybe the learning rule would minimize that disparity. So instead of maximizing the objective function we had before, which was just the covariance of the two projections, I'm going to write the objective function in terms of the in terms of the covariance of, well, rather the square of the error in the between the two projections. So the WXX is the first projection, WYY is the second projection. The difference between them is the disparity shown here by this arrow lines. And then what we're computing is the squared sum of the squares of those disparities. Maybe if we minimize that sum, then we will solve the CCA problem. This is the intuition, but it also turns out to be exact in this case, because by doing some algebra, you expand the square and you figure out that two of the terms here are constant because of our constraints. These are equal to 1. So we can ignore them and just minimize minus the cross term. But that corresponds to maximizing exactly the same objective function we had before. Well, the reason I went through all this simple algebra is to give you an intuitive idea of what the learning rule is. Because once we write down the objective function in terms of the square of the disparities, we can easily take a derivative with respect to WY and compute the learning rule by gradient descent. Okay, so that's just the disparity times Y. This is, of course, very reminiscent of what we did in the derivation of PCA for a single neuron when we obtained the OA learning rule, where the learning rule was basically the error, remember, the reconstruction error times the neuronal activity. So each data point there pulled the W towards the towards itself. Okay, here what happens, each pair of points changes the W to minimize the disparity between the two projections, so that the two projections are as correlated as possible. Okay, now that we have this, any questions?
No questions.
Okay, so now that we have this learning rule, we can already build up a neuronal model, okay? And this is our friend, pyramidal neurons. The inputs from data set Y come to the basal dendrites. From the data set X come from the apical dendrites. And there is an additional component here, which is the self-inhibitory inputs from that input from that neuron that feeds back with the negative side. Why do I have that? Well, because I want to implement this learning rule using the kind of variables that I have here, which are the two projections, okay? But in terms of those projections, so A is WXX, that's the current from the apical dendrites. This learning rule is written as A minus B, where B is the same quantity as A, but for basal dendrites. So the tricky thing is, where do we take A minus B? And that's where this self-inhibition comes in. because we can write down the Z as the sum of A plus B minus Z, right? Because that's what happens in a neuron. You add contributions from the apical dendrites, A, plus contributions from basal B, and subtract the self-inhibition Z. So we can solve this equation, and then Z equals A plus B over 2. And then we can use that Z to write down the learning rule in terms of only one variable. For WY, we just have A, and for WX, we just have B. So what that means is that we can have a calcium plateau that is computed by subtracting the self-inhibition from A that is transmitted downwards and correlates with the presynaptic spiking activity to adjust the synaptic weights. So this is the new learning rule, right? So before, what we had in the PCA learning rule, in the OYA learning rule, we had a correlation between the input presynaptic firing rate and the postsynaptic activity. In this case, it would be Z. That was the Hebbian rule. Here we say, no, no, no. You shouldn't be correlating Y and Z, then you would just get PCA. What you should be correlating is Y and the difference between A and Z, between the projection of the X data set and their shared normalized sum. So that's the idea of pulling the two projections towards each other. And this learning rule is non-Hebbian because it doesn't just depend on Z, but it also depends on A, right? So it's communicated by the activity of the calcium plateau that exists in the neuron that the synapse connects. It's still a local learning rule, but not just the output firing rate, which would make it happy. Okay, so this is a little bit trickier learning rule. And when we got this learning rule, we like said, okay, well, you know, this sounds a little bit tricky. And then we remembered of this experimentalists telling us about their interesting discoveries about this two-compartment neurons and the strange plasticity rules that they're seeing. And it turned out that this is very similar to what they found experimental. So the big experimental finding was that the learning rules in those synapses are actually non-Hebbian, that you can silence the neuron, set output, you know, suppress its output, but the neuron would still, the synapses would still learn. And that's because they had the calcium plateau that would be correlated with the inputs. And that's why we got excited about this. So again, just deriving mathematically from a principled objective function, this corresponding to CCA, we got to the learning rules that are supported by the recent experimental observation. Questions?
No questions.
Okay. All right. Oh, sorry. These are the experiments that that support this point of view. And you know, I was really lucky actually. A few of these people were my colleagues, and you know, over the years they kept telling me all these interesting things, and for the longest time I didn't know what to do with it, and then eventually it seemed to fit perfectly into this theory. So I should also say that this particular algorithm for a single neuron that computes a single one-dimensional projection of the data, right? So this is a single channel CCA, which can be implemented by single neuron, is something that actually has been suggested before in a slightly different architecture by Line 5, and also by my group. But the big challenge in this business just very similar to PCA question, was how to implement a multi-channel CCA, right? So for canonical correlation analysis, multi-channel means that you project each of the high dimensional data sets, and they could have different dimensions. But when you project them onto the same dimensional space, but that space doesn't have to be one dimensional. It can be k-dimensional, where you compute covariance in each of those projections. And so the question is how to do that. And one would think, well, the answer would be similar. You use k neurons, and in parallel, those neurons would compute this canonical correlation components. But the problem, just like before, was at how to ensure that those components are different, how to implement competition with that. And that problem becomes much harder in CCA because you have to figure out what kind of output you need to do that. And it took quite a bit of effort, but we were able to solve this problem. And so this is now the multi-channel CCA formulation, as a statistician would have it, where X now is a data matrix where vectors X are stacked at different time points. And the projection vectors are also stacked together to form a matrix Vx and Vy. And then we need to have a trace here because we need to sum over all the different components. And again, there is a normalization constraint, which prevents Vx's and Vy's from just getting big to maximize this function. But again, this is exactly the same problem. If the dimension of, if k is 1, then we get exactly the same problem as before, but it can also work for large k, for any other k. And so this is the mathematical formulation of multi-channel CCA. Unfortunately, if you use this objective function, you cannot really derive a biologically plausible network. So we had to put in quite a lot of work to guess an objective function that, when optimized, leads to a biological network. And again, the similarity matching approach came in handy because it's now like a tool. We now have like a toolbox, and we use different tools from it. And one of those is the similarity matching. And when we use the similarity matching approach, we were able to derive a network like this. And the cost here, again, is in introducing this additional population of neurons. Just like in the clustering problem, we had this interneuron, which is inhibitory, that used to be called Z, now it's called N. And that neuron implements a Lagrange multiplier, and it mediates the competition between all those neurons that represent different channels of the canonical correlation analysis. So just in the case of one neuron, you get one data set supplied to the apical dendrites, the other data set supplied to the basal dendrites. The calcium, the inputs are being summed separately, and then the calcium plateau communicates those sums, and the neurons outputs the a sum of the apical and basal, which is then being fed back by K interneurons back into those pyramidal neurons. So this network actually does compute canonical correlation analysis for multiple channels. But what is very interesting for us is that this network actually starts to resemble the cortical circuit to quite a few details. So this, as I said before, the fact that the inputs come to the two parts of the dendrites, the apical and the basal, is taken from biology, but also the existence of those interneurons. That's actually what happens in the brain, and that's the kind of the microcircuit that exists here. Again, I don't want to oversell. This is a major oversimplification of what happens in the cortex, of course, but at least we are able to, follow the same kind of approach, normative approach, starting with the optimization function and deriving this kind of more biologically plausible circuit. Again, this is the approach we followed, and this is the objective function I have. I don't expect you to be able to absorb it. If you're interested, you can look at the paper. It's actually just come out in neural computation, and we try to explain how this all works. But just one thing that I said, but I didn't have an illustration before. So this N is the activity of the inhibitor interneuron. And as I said before, it acts as a Lagrange multiplier on a constraint that enforces that the spectral the spectral radius of Z is not greater than 1.
There is a question.
Yes. Was your 6-layer network invariant to affine transforms of the point cloud you try just clusterized spirals? I don't know. It's a good question. I mean, it depends what you mean by invariant. So the network would work. The network would learn. Okay. So if you performed an affine transform of the data and through that data again in the network, it would be able to again learn to disentangle them. But I think what you're asking is, if I trained the network on the spiral data set, and then I tested it on the affine transformation of the data set, would it work? That is not going to happen if the affine transform is really big. And that's exactly why we think that this network is not a complete model of what happens in the visual cortex. So we think that the visual cortex actually is able to learn those affine transform, which this network cannot in the way that I think you are asking, right? So is that what you're asking? Okay, great. Great. Yeah, that's a great question. That's exactly the kind of consideration that made us think that we need to invent something else. But what time do I have? 15 minutes. Yeah, I don't think I have time. I can briefly mention, but I don't think I have time to talk about this. Okay, sorry, but to return to this, to the CCA network, at least mathematically, this is where this interneuron comes in. That it's a Lagrange multiplier that limits the spectral radius of Z, which enforces it's a relaxation of the constraint that normalizes the energy in the components in the CCA. So that's where it comes from. Yes, okay. So the optimization problem becomes min max rather than just mean, and that's why we have a different family of neurons, the interneurons, which actually also corresponds to biology, which makes us very happy. Okay, so this is just an illustration that our algorithm works, and in fact, it's rather competitive. For k equals 1 for one output component, we do just as well as the best algorithm in the machine learning literature, which is not constrained by biological plausibility. But for large K, there aren't really that many algorithms around, and basically our biological inspired algorithm is the best, as at least to the best of our ability of running other algorithms. I think we are doing really, really well. So this is again a situation where we started in a handicapped position by trying to respect biology, but in the end, the algorithm that we derive is rather competitive with general purpose machine learning algorithms, which were not tasked with respect in biology. So that's what gives us confidence that our approach has a bright future, that we will be able to come up with algorithms that can outcompete the backpropagation driven deep learning networks of today. Okay, so in a way of a summary to this talk and what I told you yesterday, so we were able to develop a mathematical framework that is based on optimization, where we start with an objective function, and then we derive learning rules and algorithms that contain learning rules and dynamics that maps onto neuronal networks. And none of this by itself gives you a complete picture of how the brain works. But the beauty of this approach is that there is a very rigid correspondence between the mathematics, the optimization problem, the terms in the objective function, and the form of the learning rule and the neuronal dynamics. And so if we see that our learning rules and the dynamics we derive corresponds to experiment, great. If we see that there is a mismatch, we drop it and we search for another objective function that gives you the right learning rule. But this link between the terms in the objective function and the learning rules or other biological observations is what allows us to go forward, because that provides a connection between biological observations and computational functions, computational tasks that each optimization problem solves. So this is an overview, and as the last slide, I was very lucky and privileged to work with truly brilliant scientists who helped me to push this program forward. This is Cengiz Pelivan, Mariana Tepper, Alex Genkin, Anirvan Singh Gupta, David Lipschitz, Victor Minden, Nikolai Shapashnikov, Tao Hu, Yanis Barun, Sivaj Golkar, and many others that did not participate in this particular project, and so they're not on this slide. So thank you very much for your attention.